everyone to Genetic Alliance's new webinar series. This series is called Managing the Mass of Measures, Real People's Real Data Made Useful. I'm Sharon Terry, President and CEO of Genetic Alliance, and this year we want to look at measures and standards and instruments and validation and common data elements, lots of things that in the past we haven't paid a whole lot of attention to, but certainly for Genetic Alliance as we move into translational science, and especially in the form of PCORNet, which is the nation's clinical research network, we're looking at how do we share data well and what tools are available to us. And throughout the year, we will be looking at a number of very interesting tools. I'm very excited today, particularly, to introduce Phenome Central to you all. And to do that, we're going to have Mike Brudno, who's from the Center for Computational Medicine at the Hospital for Sick Children, speak to us about it. I found it a very fascinating tool, one that I think will be very useful to especially rare diseases, but even common, and begin to be the interface we need between understanding the genome and the phenome. So Mike, you can take it away. I'll just remind everyone that you are all on mute, and what we ask you to do is to type questions at any time throughout the presentation in the little box that is appearing usually on the right-hand side of your screen as part of the setup for GoToMeeting. You can put your question in there at any time. What we do is gather the questions throughout the talk, and then at the end, Mike will leave plenty of time to answer questions, and if he can't answer them, he has a few folks with him that can help him with those. We're open to any and all questions. We really want you to understand what he has to offer. This is recorded. We archive it, so you will have access to both Mike's talk as well as the slides and the questions and answers at any time after this presentation, since I believe this will be quite useful to everyone throughout the year. And without further ado, Michael?
So thank you, Sharon, for the great introduction. I'm really excited to participate in the webinar and to present Phenome Central, which is something that we've been working on, for the last year in its current form and for actually several years in some previous incarnations. And it's really about enabling matchmaking for unsolved disorders, trying to shorten the diagnostic odyssey for many of the patients with rare, extremely rare disorders, which are very hard to diagnose clinically. So as you said, I'm from the Hospital for Sick Children at the University of Toronto, and Phenome Central was developed as part of the Care for Rare project, which is the Canadian rare disorder sequencing and analysis project led by Kim Boycott. So to start off with, I want to start by talking a little bit about clinical phenotyping. So, people are very worried about the genome and the huge amounts of data that the genome generates. But to some extent, the genome is the easy thing to capture and to analyze because it's in a very standardized form. It's just a, if you will, 20-some strings over the ACGT alphabet. So from a computer science perspective, it's easy to store. It may be a lot of hard drives, but it's easy to to understand what a point mutation means. Phenotypes are much more complicated. It's very difficult to describe what a clinician sees in a patient. Some things may be simple, like missing bones, but others are going to be quite soft markers. So, for example, when you talk about intellectual disability, or what's really the borders of mild versus moderate versus severe, or what is really a dysmorphic face, and where does whether somebody has what specific type of dysmorphism do they have. And when it comes to describing patients, we actually get very little information about phenotype. There are really two alternatives right now for describing patient phenotypes. So there used to be two alternatives up until a couple of years ago, either free text or checkboxes. When people describe patient phenotypes using free text, there are all sorts of different terms possible for the same exact thing. So dysmorphic features may be described in the clinic note as just DF or dysmorphic faces or dysmorphic features. It can get actually worse. For congenital malformations or anomaly, from sick kids clinic notes, we were able to get all of these different terms, which really mean the exact same thing. These include things like abbreviations, using the term abnormalities versus anomaly versus malformations, just typos, things like congenital abnormalities, and while all of them under the hood mean the same thing. So if you hope to have databases talk to each other, you really need to standardize these phenotypes. You cannot have every person write whatever they want to describe a particular patient phenotype. So, so all of these are terms which were actually lifted from SickKids Clinic notes. And for describing a certain patient, we may get a string which looks like this: DD, Kong, Mel4, behave pro. And for a human, this makes total sense: developmental delay, congenital malformations, and behavioral problems. But a computer, the string is completely meaningless. Other things like, DF less than delayed puberty, actually less than is likely a comma just which has been mistyped. The computer has no way of knowing this. So the alternative is checkboxes, where we standardize the description and allow a clinician to check a limited number of phenotypes, where they say, well, the patient has global developmental delay, or mental retardation, mild, moderate, or severe, and other things. But obviously, you're limited in the number of checkboxes that you can put on a single page, which is pretty much the extent that's possible within a clinical workflow. And in reality, many of these terms have extensive subterms and sub-subterms, which could be relevant for describing a patient, but you can't ask a huge number of questions on the same on one page. So we really saw that both of these alternatives are insufficient. So there is the problem that the phenotypic descriptions that are good for humans don't make sense for a computer. The fact that multiple terms can describe the exact same thing. And as a result, what we noticed, most genotype-phenotype databases store little information about features actually seen in patients. You have a lot of genotype data, it's great genotype data, but you don't know what really happened to the patient. So databases don't talk to each other about phenotypes. To answer this, what we've done is we have leveraged ontologies. And ontologies are basically connections between concepts and terms. They help describe things like there's fibrillations. There's two possible fibrillations, can be muscle fibrillation or ventricular fibrillation, and that they're both not the same thing. And they also help relate, help define each term well, and help build relationships between terms. In particular, we're using the human phenotype ontology, which has over 10,000 terms, which are organized from most general, so organ abnormality, cardiovascular abnormality, down to specific types of cardiac atria abnormality, and at the very bottom is very specific kind of phenotype, like an atrial septal defect. The great thing is these phenotypes are all not just linked to each other in terms of having this hierarchy from general to specific, but also they're linked to OMIM, so the main database of genetic disorders. So from a particular set of phenotypes, it's possible to ask how similar is the set of phenotypes to a specific known OMIM disorder. Because let's say Noonan syndrome presents with both of these, and so it's a great match for these two terms. Opitz syndrome has hypertelorism, but has a telecanthus rather than downward standing palpebral fissures, which are both abnormalities of the eyelid, but not the exact same term. So using this similarity, it's possible to develop algorithms to really assist the diagnostic process. So what we did with that is we built a system called Phenotips, which is meant to enable the use of the human phenotype ontology patient side. So the idea being that because ontologies are large, so HPO has 10,000 terms, you can't really expect a clinician to do 10,000 checkboxes and for each patient. At the same time, the best time to phenotype using the ontologies is during the patient visit. So we have, the goals of Phenotips were to make deep phenotyping simple and make it faster than paper. I'm not sure if we've completely succeeded in the faster than paper part, but at least it's a matter of a few minutes per patient to actually add all the phenotype terms that are relevant for that patient. So the key features, how it works, there is predictive search with synonyms, so you can rapidly enter the phenotypes that you actually see in your patient. You can select negatives to indicate that a certain phenotype is absent. You can add measurements such as height, weight, eye distance, arm length with automated selection of relevant terms. And the built-in diagnostic assistant suggest a list of differentials to consider, as well as an exam assistant, both of which I'll show you shortly. And these are just a couple of the screenshots. There's on the left a set of checkboxes representing the most common phenotypes that a clinician may see. But there's also the ability to do a search in the box above. For every phenotype, you can add extensive detail. You can enter measurements and see growth charts, and also see whether certain values are abnormal or normal. So we've built Phenotips, which was the a database for storing for capturing phenotype data. But one of the questions with which lots of clinicians came to us is, okay, I have this really interesting patient, and what I would really like to do is share the data with other clinicians to find out if somebody else has a patient with the same set of phenotypes, because then we can actually sequence both of them and identify the genetic cause for the disorder in both of the patients. And this was how Phenome Central was born. It was the goal was to enable data sharing from standardized data that you may be able to capture with a system like Phenotips, and connect clinicians and scientists who have access to this to the patient data, so that they can all see data which is coming from different directions. So this is the Phenome Central. It's a concept. It basically a matchmaker. It lets you know about other similar patients to the patients that you have entered yourself, and connect with users who have entered those patients. One of the key things that went into designing Phenome Central is thinking carefully about the privacy of the patient. So we thought hard about how can we enable it so that we can, on the one hand, let people find these similar patients, and on the other hand, not reveal patient data unnecessarily to a very large number of people with accounts. So to meet this need, we have decided on 3 levels of privacy for each record. Each record can be public, which means anyone can see the record, and this would be appropriate, for example, for a case which a clinician has, received full consent to share broadly to include in the publication, things like that. Private, which means only specific users or consortia can see that record, and nobody else will ever know about its existence. But also, they can be matchable, which means that the record cannot be seen as in private, but can be discovered by users who submit similar patients. So while the record is not directly observable, when a second similar record is created, the user of the second record will be able to get some limited information about the first and make a contact with the person who created the first record. And I'll show you how that works in a few minutes. So the way the Phenome Central works is you start by adding a patient, so using basically the Phenotips user interface. You do a selection of positive and negative perms. You add a VCF file or a list of genes, which we need some time to process, so that part does not happen online. And then you set the permission level for this patient. And as a result, once you've done this, for each patient, you get to see right at the bottom right here a list of other patients who share some of the phenotypic features. And for each one of them, we do a similarity based on this human phenotype ontology and create a view which shows this phenotypic similarity as well as the genotypic similarity. So it shows the fact that here are the phenotypes that match, and here are the genotypes that potentially cause the disorder. And in this case, it actually is the SR-CAP gene, which causes the floating harbor syndrome, which is where the 2 patients come from here. Once you've identified a patient who looks interesting, you can actually send an email right inside the system to the submitter of the first case in order to capture that, in order to see how many, to see if you want to collaborate to share additional data. So one case where Phenome Central was used to identify actually the diagnosis of a patient was in the case of an MFDM patients. There were 4 which were sequenced within the Forge project, which was also run in Canada, and F2 was identified as the causative gene. But there were 2 additional patients who were seen at about the same time at the CHIO, at the hospital in Ontario, in Eastern Ontario, in Ottawa. And the 2 patients had similar presentations to MFDM, but there were some things that were off. One, for example, did not have microcephaly or developmental delay, but had 11 pairs of ribs and other vertebrae issues, while MFDM patients typically have microcephaly and have mandibulofacial symptoms. The other one had extensive additional abnormalities besides MFDM, including thumb, cardiac. When we entered both of these patients into Phenome Central, they actually clustered very nicely with all of the other MFDM patients, indicating that it was quite likely that that these 2 patients also had MFDM. And upon sequencing, we noticed that both actually had mutations in the F2 gene as well. So these are also MFDM patients, but just with somewhat non-standard presentations of the disorder. And I think with rare disorders, as we're seeing more and more patients and sequencing the genomes of more and more patients, we're realizing that a large number of the phenotypic spectrum of many of these disorders is not really fully understood and really needs to be explored more deeply. So in Phenome Central today, there is extensive data from Care for Rare, the Canadian Rare Disorder Sequencing Project, the NIH Undiagnosed Diseases Program. We're getting data from RD Connect, which is the European Rare Disorder Coordination Project, and Care for Rare Australia is also participating in the Phenome Central Consortium. We have about 260 extensively phenotyped unsolved cases, mostly with exomes. And we have about 90 to 100 rare disorder scientists and clinicians who have accounts on the system. So let me now actually go and switch to a live view of Phenome Central to actually illustrate how something like this may work in practice. So I'm actually here, I will be logging into a copy of Phenome Central, not to make actual changes to the real database. So I log in and I username and password. Hopefully I get it right. And here what I see all of the data about my cases, my data. I see public data. These are cases which everybody can see. And then there are cases that are shared with me, but which are not necessarily public. So I actually have access to quite a few cases here which are not public. And I can go in and make a new patient record right here. One of the things that's very important that at the very top of the Phenome Central page, I say that I confirm that the data is entered corresponds to a real patient and that this patient has provided consent to upload this data to Phenome Central with understanding that the data is actually de-identified. Here I can modify the permissions. So right now it's owned by me and it's matchable and shared with 0 collaborators, but I can modify this. I can, for example, add a collaborator. So for example, I can say Tyler Hartley is going to be my collaborator on this record and can view and modify data. I can add a whole group. So for example, I can easily just say Care for Rare, for example, as a group for who may be able to, everybody in the Care for Rare consortium will be able to view and edit this data. Here I can also specify whether the gates should be hidden, private, matchable, or public. And I can, for example, keep it as matchable. And explains the ownership. So let me update it. The identifier field is for your own internal identifier, so I can say this is patient 123. The date of birth for the patient, and for example, male. Broad indication for why the patient is interesting, why they believe that they should be in the Phenome Central database. You can write down the family history. So you can, including ethnicities, so Irish maybe on the mother's side, and including African Americans. These are all choices which are generated from a collection of ethnic groups that we have curated. Other health issues in the family, evidence of consanguinity, no global mode of inheritance if it's known. You can then write down the prenatal and perinatal history, so including things like assisted reproduction, Apgar scores, weight for age. Then you can get into the medical history, which is the global age of onset for the condition. And finally, the measurements. So where we say it's recommended that weight, height, and head circumference is recorded, so we can go in and the patient is 6 years. We enter maybe a weight of 30 kilos, and you right away see that this is 99th percentile. You get a growth chart, maybe a height of 120 centimeters. Arm span, sitting height, head circumference of 35 centimeters, which is going to be very, very, very small. Chart small, so let's make it a little bit larger. So it does not appear because this texture chart only goes to 36 months. And finally, you get to the interesting part, which is the clinical symptoms and physical findings. One thing to notice here is we already have microcephaly selected. So that's the fact that the patient has smaller than expected head size. And then you have on the left side the checkboxes, which allow you to specify, for example, visual impairments like cataracts and nystagmus. For all of the terms, it's actually possible to unroll them and drill further down to be more specific terms, and this uses the human phenotype and ontologies, so you can drill down further and further. But instead of searching within this hierarchy, it's possible to just quickly type, for example, if you want seizures, just type that and it will find febrile, simple, complex, and then more general seizures and so on. It's also possible to type things if it's it's insensitive, I would say, to misspellings. So you can type seizures without the E and it will find them, or you can type in, let's see how badly I can mangle microcephaly, myrocephaly, and it still identifies it properly despite the huge number of typos. It even supports some number of abbreviations, so you can type CHD and get malformation of the heart and greater vessels, which is shorthand for that is congenital heart disease, which is what CHD stands for. Or so you can specify the phenotype. So, for example, if a patient presents that with microcephaly and maybe we'll do developmental delay. So global developmental delay, gross motor delay, gross motor development, maybe absent speech. First of all, the description is, as I describe more and more terms, I get an indication of how informative the description is. One of the other things that the system does is it also gives you a suggestion of other phenotypes that are correlated with the ones that are already entered. So, given these are other ones are worth thinking about as helping to refine the phenotypic description. So maybe muscle cramps are also relevant, and this will lead us to recalculate the sets of other phenotypes that may need to be investigated. At the very bottom of the section, there is a diagnosis, which basically gives a differential diagnosis given the set of phenotypes that was selected, and shows the OMIM disorders that match the specific set of conditions. You can check one of them off to be actually the diagnosis, or you don't have to select any of them. For each one, you can actually click on the link to open it up and OMIM and to see what the disorder actually is. So given that we've made this patient description, the one last thing I want to show is for every one of these phenotypes, it's possible to add detail, including to specify the age of onset, pace of progression, any additional comments, and attach an image or a medical report if they're needed to really understand the condition. One thing important, all of the data should be de-identified before it's uploaded into Phenome Central. So once I save this data, I get information about other patients who are phenotypically similar. So these are patient profiles contributed to by other users, and these at the bottom here are OMIM disorder prototypes. So basically, simulated patients generated from specific OMIM disorders, and we keep them separate. So, for example, patient 009 was contributed by the admin and is undiagnosed. You can show phenotype similarity. Because I don't have the right to see the admin's this particular patient, it does not. So I will show you this in a different view. You can see the phenotypes that are in my patient and the phenotypes in the other one. So switching to, this is the actual Phenome Central database, so I don't want to create new users here. If I look at a specific record, and in this case, there's no similar phenotypes, no similar patient profiles otherwise. Sorry. So this is one of the floating harbor patients that was contributed to Phenome Central by the Care for Air Consortium. And once this field loads, you will be able to see the similarity to other patients that are in Phenome Central. There we go. So you can see the phenotype similarity. You can see these are the phenotypes present in my patient. These are the phenotypes that are present in the other patient. There's no genetic data for one of these two, so you will not be able to see that. But if we go to patient 12, you might know has phenotype data, and look for similarity here. You can see here's a phenotypic similarity and genotypic one, and the SR-CAP gene is shown as the most likely one to cause the disorder in both of these patients. So it's as a result, it's comes up top of the gene matching. Once this is done, you can, if you want to connect to somebody who submitted one of these records, you can actually use the similarity. Sorry, 1 second. You can use the tool built into the database to connect, to directly email the submitter of the other case and to establish a direct connection. So this gives you a very brief overview of how Phenome Central works. So here there's the link to connect. You can write an email disclosing information about your patient, ask for mutual view access to the two similar cases. And if the recipient accepts, they gain view access to your case and you gain view access to theirs. So this is built into Phenome Central. So this gives you a rough idea of how Phenome Central works. I want to now go back to my slide just to give the acknowledgments. This is the hard work of the Phenome Central development team, including Marta Girda, Orion Busky, Sergio Dumitru, and others. And at Care for Rare, Kim Boycott, Sarah Sawyer, and Tyler Hartley have helped us a significant amount on the clinical side, as well as our collaborators at the NIH UDP and within the HPO and Monarch group. And I now would be very happy to answer any questions and to also walk through the the workflow, show other features of Phenome Central that I may not have highlighted now. Thank you.
Great, thanks very much, Mike. That was really terrific. And we do have a number of questions teed up, and I'll take them more or less in the order that they've been given to us. I'm going to group like questions with each other. So the first question is Phenome Central available for public use? And if not, how is it accessed?
Yeah, it's a great question. So what we have done with Phenome Central is we give account. You can apply anybody. So we ask that the people who apply for accounts, and we only give accounts to clinicians and/or scientists who are affiliated with a public or nonprofit institution and can share their patient data through Phenome Central. The restriction is, unfortunately, we wanted to, at least to begin with, control who can add data in order to make sure that the data stays of high quality. We do very much envision in the near future, we're hoping, expanding Phenome Central to actually include patient-contributed data, but we have to be very, very careful about things like privacy and things like making sure that we understand what part of the data is clinician-contributed and hence likely to be more accurate versus things which are patient contributed and where there could be inconsistencies.
Great, thank you. And I'm going to actually follow on a question that comes a little later, but I think is relevant right now, and that is that some have said that Phenotips and Phenome Central is not really easy to use in the clinic because it's too slow. What do you have to say to that?
Phenotypes, we have never heard the complaint that it's too slow. If it's, it takes, entering a patient takes about 5 minutes. For clinical genetics, that's not too slow. For, obviously, for the encounter with your general practitioner, that possibly is a bit slow, but then there are likely many fewer things that the general practitioner would want to specify about phenotypes. I would, whoever asked that question, I would love to find out why they believe it is. It's, whether it's a software that's slow or whether just the whole workflow is too slow. And so I think it's a great question, but I think I need to understand more about what is the issue.
Right, good. So what you'd recommend then, of course, is that people give you that kind of feedback because that's very
Yeah, no, I mean, we all feedback that we get, we follow up on. Everything, all suggestions are added to the development queue, and especially if we hear from multiple people that the same features is what's necessary for Phenome Central, for Phenotypes to be, or Phenome Central to be adopted by this following group of clinicians, we would probably try to do it as quickly as possible. Address those.
Super. Okay, a couple questions on informed consent. One question asked, does Phenome Central require that the patient sign an actual consent form, or do they just give their verbal permission to upload their data? And then a second follow-on question regarding informed consent from patients, do they sign an IRB-approved consent form? So similar questions there.
Yeah. So what we have done with Phenome Central is we ask that the clinician has received consent from the patient to upload the data to Phenome Central. Obviously, the form in which this consent takes place is going to be variable across different countries, different institutions, and the form. In some places, just a permission from the patient, that country may consider sufficient, while in other countries that's considered, that the written consent is needed. We really leave this with the submitter of the data. As long as they have followed whatever are the necessary regulations on their side, we consider that sufficient. The data is de-identified, needs to be de-identified to be contributed to Phenome Central, and we do have somebody who regularly checks the data to make sure no identifiable information is included. And so we try to address it on our side as well. But we do not have a specific, consent form that needs to be filled out for Phenome Central specifically. What we do instead is we have written parts of IRB applications for other institutions where they indicate that they will upload the data in Phenome Central as part of some other research project.
Great. And you fall, like we do with our peer project, in a place that IRBs right now don't know quite what to do with, and neither does the human research protections people, since it isn't really a research project per se. It's more a service tool and also a way for people to be discoverable to one another, and that's a little different than a research project.
Yeah, this is, I mean, this is obviously a big problem with a peer tool or with us, that, IRBs don't really understand, what to do with things which don't look like standard research projects and don't look like, it's not really clear on which side of the identifiable or non-identifiable data they fall.
Right, right. Good. Another question, is there any data about the medication that patients are on, including, for example, past history or concomitant medications, et cetera?
Yeah, that's a great question, and this is something that we're interested in adding, but it's not something that's available right now. It's very much on our to-do list, especially as part of opening up Phenome Central in some way to the actual patients who are being described, rather than just to the clinicians who are treating them.
Great. And I should say, you can all expect that, of course, Mike and we are talking because our two tools look like they are great complements for one another. Next question, how about a business model and/or installing and using Phenome Central for commercial use?
It's again, all great questions. Business model for Phenotips, which is the sort of the data capture component which is under the hood of Phenome Central. We actually there is a company of which I'm a part of, so full disclosure, which provides user support for Phenotips and installation services. The software that actually goes into it is completely open source and free. Anybody can download it. Anybody can use it for pretty much whatever purposes, as long as they abide by the open source license that we're using. For Phenome Central, I'm certain that there is a business model to be explored for all of the data. However, I think before we go that route, we need to be very, very comfortable with the fact that the patients have undergone a much more thorough consent process, and that they're truly aware that this data could be, going to that the underlying data could be used for, for things besides the diagnosis and potential treatment.
Great, thanks for the answer. Next question. The 4 stars that you showed, do they represent the percentage of phenotypic expression, or how is the 4 stars calculated?
Yeah, it's a great question. So the stars are basically a measure of how unique one would expect the patient, such a description of a patient. And when I formulated this to the genetic counselor who works with us, I said basically, well, 1 star means that there's somebody who works at our research institute who is like this, and who are just a little bit strange, but probably not very rare. And 4 stars means that there's 1 case like this in kids a year, or 5 stars means that there's basically that you expect to see 1 of these in a lifetime. In reality, they're computed by looking at the phenotypes that are selected, looking both at the rarity of the actual phenotypes among different rare disorders. So a phenotype which is present only in a small fraction of disorders, all of which are very rare, is going to give you make a higher contribution than phenotypes that are very common, like microcephaly, for example, is a reasonably common one. At the same time, and also, if there is broad coverage, so for example, there are many body systems which are impacted, that's going to give a higher score than phenotypes that are that only come from a single body system. So we're looking both at the rarity of the phenotypes and the fraction of the body which the phenotypes actually cover.
Great. Can the information be transferred to electronic health records, or does that require double entry on the part of the clinician?
So that's another great question. So electronic health records in their current state are not as good for supporting extremely structured data from a very rich ontology such as the HPO. We are actually working with several hospitals to implement phenotypes clinically. So in which case phenotypes basically becomes part of the medical record and the clinician would document the patient in phenotypes, which then creates an abstract, which is recorded in the electronic health record, and then can also be from Phenotips pushed to Phenome Central by clicking a couple of buttons. This integration is still something that I think is a bit tricky, but something we're hoping to accomplish with several hospitals, I think, within this year, within 2014.
Okay, and to follow on to that, then do you synchronize regularly with changes from HPO and OMIM, for example?
Yeah, so the reindexing of HPO is very trivially done within Phenotips or Phenome Central, and we do resynchronize on about a monthly basis.
Great. So there are many attempts to capture patient data in separate databases. Wouldn't you agree that a consolidated or federated sharing of databases is best given the rare allele frequencies? And then a follow-on question to that, how many samples are in Phenome Central?
Yep, so again, there is a project going on right now called the Matchmaker Exchange, which includes Phenome Central, the Decipher database, and Gene Matcher, which is a database developed at between Harvard and Hopkins, and all of which store some number of rare disorder patients and their genetic data and some phenotypic data. And so we're right now working, actually, we've built an API which allows us to exchange data, and we're hoping to be able to actually have a federated model built into Phenome Central and Gene Matcher and eventually Decipher, with the first parts of it live possibly within this month.
Great. And for our listening audience, we will be having webinars on Decipher and Gene Matcher both. So stay tuned and look at our website for those dates. Another question, how does Phenome Central interact with REDCap, and does it interact with any EMRs? I think we asked, could it push information to the EMR, and you rightfully said that the EMR doesn't accept structured data so well. But I guess the question is, can it draw data out of the EMR if it can find the right fields, I would imagine, and can it integrate with something like REDCap?
Yeah, so with REDCap, we have not really looked at any integration at this stage, but I think we will be talking about that with you, because a lot of the systems that the Genetic Alliance develops are based on REDCap, so that's certainly going to be on our agenda. We are working on a system which will take a clinic node and try to identify the most salient phenotype terms from that node. And stay tuned for that. My guess is, within a couple of months, we'll have something available where you can upload just a textual record, and it will try to parse the terms.
Super. Then a question with potential chemical, biological mutations. Would you think you would start to see any emerging or regional biological changes in the subjects that are being entered into the database?
I'm not sure I fully understand that
question. I'm not sure I do either. I think they might be asking whether you could see some kind of trending. So, for example, if we start to see, and I'm going to sort of make this up because I'm not exactly sure, but if whole exome studies start to show pockets of children with certain autism spectrum disorders in a certain area, would you be able to start to see those trends?
Well, I guess you would be able to see easily see phenotypic trends if the patients are sufficiently described in sufficient detail by going to, by building things like this, which only we can do because nobody else can access all of the data in Phenome Central, but see the similarity between the various patients and see if there are some natural groupings. But I'm still a little, I'm not really sure if Phenome Central can be used for that purpose. The goal was really to enable the discovery of additional patients with a specific rare disorder based on the description of a single patient.
Okay, yeah, and actually the person elaborated a little more. For example, Chernobyl or Japan, you know, if we start to see genome changes based on the radiation exposure, theoretically you could start to pick that up in a system like this.
I think only theoretically. In practice, the geographic diversity of the patients in Phenome Central is quite high, so it's quite unlikely that we'll have a very large number of patients from any of such grouping, unless there's a very large number being entered, which maybe, I guess. But besides a few types of cancers, I can't imagine that would be too prominent.
Right. Okay. Let's see. Can you say the name of the data sharing federated model again?
This was the Matchmaker Exchange project.
Right.
And this includes Gene Matcher and Decipher, us, and several other groups.
Super. Technical question. Can you use Phenotyps with Oracle rather than HyperSQL?
Yes, you can use it with any SQL-like database.
Okay. And then have you had conversations with the National Library of Medicine or NCBI at NIH?
We have, I guess the question is for which specific purpose. We've had multiple conversations with NHGRI through their UTP program and with some other people at NIH, but not specifically with the NCBI. I'm not sure exactly why, but what the person has in mind in terms of their question.
Yeah, and I think they were heading toward data standards, and in fact, I'm just fresh out of a Phoenix meeting, and Phoenix is the standards validated instruments that NHGRI, National Human Genome Research Institute, is working on, and then there's PROMIS, which are the patient-reported outcomes, common definitions, the common data elements.
Yep.
As you were going through your presentation, Mike, probably because I just stepped out of 2 days of those meetings, lots of the things, of course, that you're capturing are things that can be captured through validated instruments. And I guess the question's asking how much of those kinds of instruments are integrated, or is that the kind of thing that, in fact, our marriage with REDCap and the standards that we're putting into REDCap would, in fact, help with your system?
I think so. For the human phenotype ontology is very broadly used in the rare disorder community, but I think a bit less so outside of it. So I'm not sure what fraction of things like the HPO would be part of common data elements that are stored by some of the more common disorder-oriented efforts, such as some of the common data elements ones typically are more oriented towards that. But I think any kind of work to standardize the kind of data that's captured would be great.
Super. And in fact, one of the pieces of the conversation over the last 2 days that I was in is that we are setting up a rare disease work group for Phoenix. So we'll certainly, be in touch with you and all the other projects to see, of all these ontologies, how do we start to bring them together so that we're making the best use of the community's knowledge.
Mm-hmm.
And we have about 10 minutes left, and I'm going to put you on the spot a little because I didn't actually ask you this ahead of time, but I wonder if you could show the family tree building that you have.
Oh, sure. I'll show that in a slightly different server, but this is
sorry that I'm putting you on the spot a bit here.
That's okay. I'm happy to do that. So this is the Phenotyps playground. So this is where people can go to play around with phenotypes and to try to see how the system works. So if I go here and I click on the new patient record, what I will get is a form which looks very much like the HPO form, like the Phenome Central patient data form, but this is not configured specifically for HPO. Under family history, there is the draw pedigree function, so I actually make the patient a female just to make it fun, and draw pedigree. And here it is. And I can click on, I'll start with a single proband. So this starts with a single node. To add parents, you just click on this node. And there we have parents. You can add a spouse for the mother by saying here, the mother now has two partners. The partners, they had a child, I think that was another boy. You can indicate what phenotypes the proband has by just typing here, and it'll give you anything from OMIM Angelman syndrome, for example. Maybe there is something else. And this gives you a key at the top, which lets you highlight which patients have specific phenotypes. You can add more. Let's say, for example, let's say this is actually related as a child of this marriage. So basically, the woman has married two brothers, I guess, in order. You can specify that this specific marriage is over. So, and if you want to indicate that, for example, somebody else had Angelman in the family, you can just highlight it this way. You can add additional, siblings. So this adds a sibling, and this will, or you can add a third spouse and a fourth spouse, just to show that it can work. It's really a flexible pedigree drawing tool with all kinds of data. One of the things that we're going to do is make sure that each of these can be a separate Phenotips or Phenome Central entry. But for now, it's all tied around the proband, which is the one highlighted in bold right there. So this is a quick demo of the pedigree drawing functionality in Phenotips, and also in Phenome Central. Actually, you can draw the pedigree there as well.
Yeah, it's really terrific. Those of you who saw that I didn't mute my microphone, I said, I want this. I do. I think this would be, again, a fabulous addition to some of the stuff that we're working on. I think people who have to walk around with crates of records for their family because there are lots of conditions within the family, or because they have a child with an undiagnosed condition, would really appreciate something like this.
Yeah.
And then last question, someone's wondering if they downloaded Phenotips and installed it locally, then are they free to change it for their own clinical use?
Completely. So one of the things I didn't have time to show, but it's more Phenotips rather than Phenome Central. Phenotips is highly configurable, so there are lots of things which you can change without actually having to go and get your hands dirty in the code. For example, if I now save this pedigree and close this window, this list for example, the common phenotypes are completely configurable from the administration menu of the database. Each person can configure them for their own use. If you want to change the code, you're very welcome to do that. It is distributed under the LGPL license, so any changes that you make directly to the code, you should be making publicly available to others, and we would be happy to work with you to contribute them to the broader open source project, because if you found it useful, odds are there's somebody else who found it useful as well. But it's highly configurable in its current state, even without having to know how to write code. But then if you do write, you can, because it's all open source, you can get the full code to it. You can do anything.
Super. And then, Mike, is there anything else that you would like to show us before we close out today?
Now you got me on the spot. The pedigree drawing was easy. I think this is just this was the main things that I wanted to demonstrate, is both more the Phenome Central, the data sharing aspect, but Phenotips is there as well. And if anybody has any questions after, if they want to try them out on their own, we're very happy to help. Just send us an email, and we'll try to respond to all of them as quickly.
Well, thanks so much. This was really wonderful, very effective presentation. I'd like to remind everyone that this is part of a series that will go on much of the year. We're still adding workshops and webinars, so that we can see that we'll be looking next at Phoenix, which is the project that I just spoke about, which is a project of NIH, especially the National Human Genome Research Institute, to help integrate genotypes and phenotypes. And then throughout the rest of the spring, essentially, we'll be looking at REDCap, I2B2, Mini Sentinel, Promise, and then BD2K, which is Big Data to Knowledge, Common Data Elements, CDISC, which sets standards, Decipher, which Mike talked about, the human phenotype ontology itself, which is this project you just looked at is based on, Transmart, and then several others that actually are in the process of being teed up. So what we hope to offer the community is an overview of the tools, the standards, the validated instruments, common data elements, metadata, et cetera, that's offered to all of us, and to start to see how these things can intersect with one another and how we can really make clinical data useful across the board, so we start understanding phenotypes better and we start to understand the progression of human disease better. So thanks again very much, Mike, for what you offered us. I'm sure that you'll get some feedback. We'll be able to supply Mike's email address to anybody who emails us. You can also find him pretty easily. And we ask you all to just register for the rest of our series as they come about. We'll keep you informed as we go. Thanks very much.